Hi, let's talk about the formed elements of the blood. In this video, we'll discuss the formed elements of the blood, their relative abundances, and their development. We'll also discuss hemopoiesis and how it's regulated, and touch on the major hematological malignancies and the tissues from which they arise. So, formed elements refers to any blood cell or large cellular fragment, and there are three major categories of these. So there are erythrocytes. Erythro means red cell, so these are red blood cells. By far the most abundant formed element of the blood. There are between 4.8 to 5.4 million RBCs per microliter. And red blood cells are very important in gas delivery, especially that of oxygen. The next most abundant formed element are thrombocytes or platelets. Platelets are typically somewhere between 150,000 to 400,000 units per microliter, and these are very important for clotting and coagulation. The last group of formed element are the leukocytes. Leuko means white cells, so these are the white blood cells. And typically, overall, a complete white blood cell count is between 5,000 to 10,000 leukocytes per microliter, although these aren't uniformly distributed amongst subtypes. So, for instance, the most abundant leukocyte are the neutrophils, followed by the lymphocytes, followed by monocytes, followed by eosinophils, and then the least abundant are basophils. I remember this order of relative abundance by the mnemonic, never let monkeys eat bananas. Leukocytes relate to immune responses, as we'll see in subsequent videos. So let's take a look at this light micrograph here. This is a stained blood smear, and what someone has done here is they've taken a couple of microliters of blood, they've stained it so that we can better visualize the cells and particular parts of the cell, and they've photographed it. So this was the original method by which someone would evaluate blood. So this is the original CBC, or complete blood count. Nowadays, a lot of these techniques are more automated, but this is a useful follow-up tool to an automated CDC. Not only does it allow people to see the relative abundances of formed elements, but it also allows them to look at the sizes of cells, particularly whether a cell is too small, meaning it is microcytic, whether it is of typical size, normocytic, or whether it is abnormally large, macrocytic. Blood smears also allow individuals to see if there is the presence of any parasites within the blood. So endoparasitic infections can often manifest on a blood smear. So let's talk about the process of blood cell formation, also known as hemopoiesis. Sometimes you'll hear this referred to as hematopoiesis. This is precisely the same thing. So hemopoiesis originates in red bone marrow, also known as hematopoietic connective tissue. Hematopoietic connective tissue is abundantly found in the spongy bone, so the cancellous, or the air spaces around bone trabeculae in spongy bone. And it consists of stem cells. Now, all of these stem cells originate from pluripotent stem cells, but as they become increasingly derived, there are two major categories here. There are myeloid stem cells and lymphoid stem cells. Myeloid stem cells, as we see, will give rise to erythrocytes, thrombocytes, granular leukocytes, that's white blood cells with granules in them, mast cells, and monocytes. Whereas lymphoid stem cells give rise to agranular leukocytes. That would be white blood cells that lack granules, with the exception of monocytes, which, as we recall, are products of the myeloid stem cells. I really like this graph here because it shows the relative importance of different tissues from which blood cells develop at different stages of development. So this is very well divided between prenatal hemopoiesis and postnatal hemopoiesis. So the red line down the middle is parturition or birth. And if we turn our attention very briefly to the prenatal portion, we can divide this up into thirds approximately. And we can see that during the first trimester, most blood products are produced in the yolk sac. During the second trimester, most blood products are produced in the liver, but we also see substantial contributions from the spleen and increasing contributions from the bone marrow, which takes over in the third trimester and persists in the postnatal period as the single greatest source of hematopoiesis. Even then, in the postnatal, we can see the differences among the different regions of bones. So for instance, the spongy bone within the vertebral column or the vertebrae and pelvis produce the lion's share of hematopoietic tissues, followed by the sternum, the ribs, the bones of the lower limb, so the tibia and femur respectively. And we can also see the dark horse here, the lymph nodes. So the lymph nodes really begin hematopoiesis about the middle of the second trimester, and then they begin to asymptote and account for perhaps 18% of the total of hematopoiesis, which is so very, very interesting. So this chart is a very succinct organization of the various developmental pathways of the formed element. So we can begin here with the pluripotent stem cell. And this is a choose your own adventure, so we can either go that way or that way, but we have two choices here. These pluripotent stem cells can develop into myeloid stem cells or lymphoid stem cells. So as these cell lines become increasingly more developed, we'll see them move through different types of cells. So for instance, at sometimes in ontogeny, these lines will have what are known as progenitor cells, and then precursor cells, and then various formed elements that we can find in the blood, and then tissue cells, which we can find either in the tissues and also sometimes within the blood. So with myeloid stem cells, there are four basic pathways, three of which are things called colony-forming units that get their name from their appearance on plates. So they kind of look like bacterial colonies. So these are colony-forming units, erythrocytes or erythroblasts, colony-forming units, megakaryoblast, and colony-forming units, granular monocytes. So colony-forming units, erythroblasts, differentiate into pro-erythroblasts, which eventually become red blood cells, or erythrocytes. Colony-forming units, megakaryoblast, become megakaryoblast. These are very large cells that kind of break down into smaller fragments, and those smaller fragments are the platelets, or the thrombocytes. We also have myeloid stem cells differentiating into mast cells, which are excellent tissue cells at degranulating histamine. And then we have colony-forming units, granular monocytes. So we have various types here: basophilic myeloblasts, eosinophilic myeloblasts, and monoblasts. I think the the root here that we want to pick up on is blast. Whenever we see blast, we should think that this is an immature cell. Blasts are often going to differentiate into cytes in various tissues. So cytes are more mature cells typically. But here, they're going to differentiate into a variety of leukocytes. So basophils, neutrophils, eosinophils, which are all granular leukocytes, as well as monocytes, which can then differentiate into macrophages. Macrophages, these are large phagous cells, so these are large eaters that can go around consuming pathogens, parts of defunct cellular machinery to break it down for the immune system. The other direction that we can travel here is to lymphoid stem cells, so we can go to T lymphoblasts, natural killer lymphoblasts, and B lymphoblasts, which will differentiate into various T lymphocytes, natural killer cells, and B lymphocytes. And then B lymphocytes, when they're activated, can become plasma cells, which are the cells that produce antibodies or immunoglobulins. So as we go through other videos, you can keep this pathway in mind as to the development of these different formed elements. Now, a lot of formed elements are under the regulatory control of various substances, and in particular, as a broad category of regulatory substances, there are these things called cytokines. Cytokines are small signaling molecules which are typically either peptides or glycoproteins. As such, these are incapable of crossing the plasma membrane of other cells, and so their action is on, surface membrane receptors of cells. So these are very important cells at modulating the immune system. Oftentimes they're referred to as immunomodulating agents. They're very different than hormones in terms of the levels in which they act and their specificity. So hormones, they typically circulate within the plasma at picomolar concentrations. At nanomolar concentrations. That's 10 to the negative ninth. Whereas cytokines circulate in much, much smaller or picomolar concentrations at 10 to the negative 12th. Now, whenever there is an immune response, an infection or trauma, these can increase, 3 orders of magnitude. So they can increase in their levels very quickly. Like hormones, cytokines can act on either the tissues that secrete them, meaning they can have autocrine action, or they can act in the tissues that surround them. They may have paracrine action, or they might have more systemic or endocrine actions on cells throughout the body. Now, originally, cytokines were described in leukocytes, but they are secreted by nearly every nucleated cell. Cytokines are also pleiotropic, which has a different meaning from the world of genetics. Pleiotropic in this instant means that it has a different effect on different target tissues. So, for instance, if we were to take a particular cytokine, interleukin 4, IL-4, which is what we can see represented over here. On B cells, interleukin 4 will activate, proliferate, and differentiate them into plasma cells. Whereas on T cells, interleukin 4 will be responsible for T cell proliferation and mast cell proliferation. It's interesting that cytokines may also be pathologically dysregulated, meaning that they can increase in numbers to the extent that they can become harmful to the body. This is a condition known as hypercytokemia. So hyper, too many, cytokine, and then emia, so too many cytokines in the blood, colloquially, this is referred to as cytokine storm. So you might have heard of this with respect to the flu or the COVID-19 pandemic. What happens here is an overabundance of cytokine can lead to an overabundance of inflammation and, multi-system shutdown, which can be deadly. Now, specific cytokines that affect formed elements are called hemopoietic growth factors, which are sometimes just referred to as hemopoietic cytokines. So these are cytokines that specifically stimulate growth and or development of blood-formed elements. And there are a couple of major categories here. Again, these are very broad strokes, but we have things called colony stimulating factors, or CSFs. Colony stimulating factors are usually going to work on colony forming units, CFUs, so they specifically are going to enhance myeloid stem cell mitosis and further differentiation. We also have interleukins, between leukin white blood cells. This is, again, a very vast and diverse system of cytokines. These were originally described and thought to only play a role in the communication among white blood cells, but we since have discovered that most nucleated cells are capable of secreting them. In particular, interleukins in the formed elements are responsible for mitosis and development of many hemopoietic cell lines. And then two that we will specifically discuss in subsequent videos are erythropoietin, EPO for short, and thrombopoietin, or TPO for short. Erythropoietin is secreted in response to hypoxia or low levels of oxygen, and it enhances mitosis and development of erythrocytes, whereas thrombopoietin is secreted in response to cellular damage, and this is going to recruit larger numbers of thrombocytes or platelets from megakaryoplasts. Now finally, I just briefly wanted to touch on hematological malignancies. Now this is probably a topic that an entire course can be devoted to, or many courses can be devoted to, but very broadly there are three types of hematological malignancies. There are leukemias, lymphomas, and myelomas. Leukemias, which account for about 30% of hematological malignancies, can come in many different forms. Typically, these are malignancies resulting from bone marrow or leukocytes. In particular, you see an overabundance of immature white blood cells, but there are also forms of leukemia where you see an overabundance of mature white blood cells that just aren't formed properly. So this accounts for about a third of hematological malignancies. More than half of hematological malignancies are lymphomas. Lymphomas are malignancies which arise from lymphatic tissues. And then a minority of hematological malignancies are myelomas. Myelomas originate from bone marrow and or plasma cells, and they are, as I've said, the minority of hematological malignancies. Now, I don't really want to talk about prognoses or anything with respect to these large categories, because they really don't mean much with respect to these large categories. Each of them can be broken down into finer, granular detail. I would encourage you, if you'd like to learn more about any of these, to visit this website, seer.cancer.gov, which has all of the data and the background that you would want to know for any type of malignancy, not just the hematological malignancies. So let's leave you on a question here, and that is, leukocytes are the product of which stem cells? Colony-forming units, granular monocytes, lymphoid stem cells, myeloid stem cells, or both myeloid and lymphoid stem cells. The correct answer here is D. Both myeloid and lymphoid stem cells are going to produce white blood cells or leukocytes. Thank you very much for your time.